Hello, this is Lecture 2 of A New Theory of Evolution. Today we're going to cover three different topics. The first one is we're going to pick up our first application of prene-theory, uh, and that's to sociology. Uh, and in particular, we're going to get into the manuscript on the origin of societies, a new theory of societal evolution, a new scientific foundation for the social sciences by moi. And notice that this is a draft dated January 31st, 2025, which is today. This is not the final copy of this article. In fact, there may never be a final copy. This is one of probably 30 or 40 drafts. And the draft date, January 31st, 2025, is sort of a best if used by date. That is, a week from now, there'll be changes to this. The same can be true, said for Darwin, Turing, Dawkins. It's a never-ending story. All right. The title is clearly audacious, taken from Darwin's On the Origin of Species. I hope Darwin wouldn't mind. And this new theory of societal evolution, I call it a new theory because there's been other theories of societal evolution going back to the mid 1800s, and in fact, preceding the publication On the Origin of Species by Darwin. The second subtitle, or the subtitle, is a new scientific foundation for the social sciences. So it is my premise that the social sciences may turn out to be just as much a hard science as biology is. And that's why I've written that there. Okay, so part one, we're going to begin to get into that book. In part two of this lecture, we're going to see a couple of videos. Part two is about bringing you up to speed on the cell and what's happening inside the cell. And the two videos indicated here will show you what's going on inside the cell. And it should disabuse the listener of any idea that the what's going on, that the cell is just a bag of goo, and that molecules sort of at random move around and happen to bang into each other and chemical reactions occur. And that's what the cell's about. This view will show that that old view is out of date and that a new view is in order. That new view being that the cell is in fact a computational device carrying out algorithms. It's very interesting. The two videos are about 10 or 11 minutes in total. The third part of the discussion is the implications of the cell and its molecules being able to compute. And so we'll talk about a number of things, including DNA computers. This third section is a sort of rather technical discussion involving a lot of theoretical computer science. And as a result of that, if that's not of interest to you, I suggest skipping the third section. All right, let's get underway. Now, in 2024, I published a book titled Darwin, Turing, Dawkins, reference Adelman, which explores a general theory of evolution. This article is a specialization of that theory to the social sciences. So it's not uncommon in mathematics to have a very broad theory of something like groups, and then you might specialize to groups of elliptic curves. Okay, so Darwin, Turing, Dawkins has a lot of stuff in it. I've tried to extract that stuff and write this shorter document, the one we're looking at, which is about 40, hang on, which is about 40 pages in length, and less demanding, I think, than the book, but still, I think, saying something worth saying. Okay, Darwin, Turing, Dawkins is available for free download at Archive. For the latest draft, see adelman.usc.edu, which is my website. Throughout this article, DTD will refer to Darwin, Turing, Dawkins. Okay. Table of contents. I'm going to read it just so you'll have some idea of what we're going to go through. There's a preface, which we just read. There's an introduction, which has two subsections, social evolution and evolution. There's a section called prenes, subsections, what is a prene, and the resurrection of smallpox. Okay, and I know a lot of you probably also already read this, but that's kind of an interesting title for a section, I think. Cultural prenes. What is a cultural prene? How to be an unsuccessful cultural prene? How cultural prenes organize people? How cultural prenes acquire people? How cultural prenes, prene sets, I've said here, program their own mutations. Then the prophet and the Messiah, the societal ecosystem, and the struggle to survive. Then the next section is mutational power, and I talk about the complexity of societies, and a title, greatness, which if you happen to be on a quest to achieve greatness, then this might provide some guidance. Members of society, cultural preens and members of society, the human brain. We'll talk about the human brain. Maybe we could talk about it a little bit now. Okay. What is the human brain, in your opinion? I mean, I know that's a pretty wide open question. Anybody got anything to say? Okay. It's a
natural computer, maybe.
It's a Dipsy decide what?
Yep. A natural computer.
Yes. How many of you think that the human brain is a computer? Okay. How many of you think, does anybody hold a contrary view as to what the human brain is? Okay.
Maybe
I
this
also,
is oh, apologies.
Nathaniel Johnson, yes.
Yes. I was thinking maybe it's a little bit more of a specialization on that. Maybe it's like a specific algorithm running on a biological computer.
Mm-hmm. Okay, that's cool. I kind of like that. Yeah, and I admit that if we want to go further in this particular direction, we need to straighten out what the software and what the hardware are. Okay, but
I'd be very surprised if the brain was a classical computer, though.
I'm sorry, Chandra.
I'd be very surprised if the brain was a classical computer.
Okay, what do you mean by classical computer?
As opposed to being a quantum computer. I don't know if that makes any sense right now.
It makes sense, and it's my understanding some people, like the famous
Roger Penrose?
Yes, Roger Penrose, who did wonderful things in physics and even in mathematics, Penrose tilings and stuff. He's going around saying, well, maybe it's a quantum computer, maybe quantum's important. Maybe he's right, but I don't see, and I haven't explored this or kept track of this particular thread. I don't see any real evidence to think that it's a quantum computer or quantum mechanics has much to do with what the brain does. Okay, that's my opinion. At any rate, I think it's a computer. Okay, and when you are born, what does that computer know about, democracy?
Nothing.
Yes, Anesha Singh. It knows nothing. It knows about as much about things like democracy as if you bought a new, you know, iPhone, say, and it just had the basic operating system in it. It knows nothing about democracy. Now, once you have your iPhone, you may download lots of stuff. If you're interested in politics and stuff and in the philosophy of governance, you might download a lot of stuff about democracy and maybe its opposite, which we'll call autocracy. Okay. But when you're born, it doesn't know the first thing about it. Okay. What do you imagine we have a brain for? I mean, bacteria don't have brains. Why do we have a brain?
To help with our survival in a complex environment.
Yes, William Weinberger. Yes, but I think it's more than that. Where did your brain come from? Why, when you came out of the womb, did you have this thing in your head, this thing we call a brain?
Because evolution, of course.
Of
Natural
course.
selection.
Evolution.
It's the basic, I mean, not basic, it's kind of the prior information that you have to come with to operate.
It's from our DNA.
I'm sorry, what?
From
Shenhua?
our DNA. The DNA
Yes.
formed us.
Yes. I mean, what built your brain? I think Shenhua's told us, right? Your DNA built your brain. It built your brain just like it built your heart, right? Okay. And the reason, of course, natural selection. By building a heart or by building two lungs or a brain, it helped us survive. That is, other evolutionary paths that didn't invest in building brains probably went extinct or may have gone extinct. Not all of them, of course, because there's still plenty of bacteria around. All right. So, yeah, it was built by our genes. By the way, are all hearts identical? I mean, if you go and take a bunch of human hearts, that's a hypothetical, don't do it. And you put them on a desk. Let's say you have 10 of them. Would they all look the same? No, they'd all look very different. I mean, very different. If they were all exactly the same as just an organ, that would make brain surgery less demanding, right? Okay. Excuse me, if all the hearts were the same, that would make cardiology less demanding. So I think I messed that up. If you took two hearts, they wouldn't look the same. Do you think if you took 10 brains, they'd all look the same? The answer is no. And I mean, just as a matter of fact, it's no. Now, what control do your genes have on your brain? Why do they all look different?
It could be both a factor of genes and also the environment.
I'm sorry, Sanpad, I missed the last word.
It could be also different because of the environment.
Yes, you're right, except that when our brains are built, we're inside a womb. Okay, and wombs are pretty controlled environments.
I think you might be referring to like a long-term environment is what I was kind of thinking of, just for evolution. So
Yes,
some
but-
people's bodies are larger because they were put under different circumstances, like over like
Yes.
hundreds of thousands, millions of years, So
Thanks. Yeah, thanks for that
I
clarification.
would say that also short-term environment. I mean, like if you have some toxins while you are developing in the womb, your brain may not really work. Okay, so there's not.
Yes, that's right.
Environment matters and genetics matter, both of them.
Good point, Luis.
So back to the genes, maybe the part of genes that produce the brain might not always be the same.
Yes,
And also, I mean, the brain's not the same, but they're pretty similar. Okay, so it's not. I mean, we all have a brain. They have like different areas that tend to have the same type of function.
yeah,
in different areas. Yeah, they're not quite the same, but they're pretty.
Okay, Jose Luis.
I just yeah.
I think you're right. But for example, these different areas of the brain, can small genetic changes say that I'm going to invest more neurons in this section of the brain that have to do with speech, then I'm going to invest in the section of the brain that has to do with just storage of information. Maybe I devote a little more to one and a little less to the other, et cetera, et cetera, et cetera. So a lot of that is under control of the genes, but you shouldn't think that all brains or all hearts are the same. Depending on the genetic makeup, more or less can be invested in some part of the brain. It's sort of like if you were building a computer, how much RAM are you going to put in? How much long-term storage, right? How much are you going to invest in the CPU? And all right. Anyway, enough about that. The human brain. And then we're going to talk about people's cultural meme sets, something that we haven't defined yet. And then a kind of interesting topic, I think. Cultural preens and emotions. How many of you have ever experienced emotions? Well, maybe I should ask how many have never experienced emotions. Okay. So, yeah, but why do we have emotions? Where do they come from? What is their purpose? And how are they used? That's a big question. So, for example, well, I'll skip the example. And then I have a sort of summation section, which the origin of societies. How should we now think of the origin of societies now that we've learned this stuff? Okay, so let's get underway. Introduction, Social Evolution. Herbert Spencer was a 19th century English polymath. He is widely considered to be one of the founders of the field of sociology. I'm going to use the text-to-speech option. So here we go.
Remarkably, he coined two phrases that remain familiar to this day: survival of the fittest and social evolution. Spencer was a contemporary of Charles Darwin, and the two exchanged numerous letters regarding research. Spencer's work on the evolution of societies began before Darwin's publication of On the Origin of Species, Darwin, 1859. In that book, Darwin acknowledges Spencer as follows. Mr. Herbert Spencer, in an essay, originally published in the Leader, March 1852, and republished in his essays in 1858, has contrasted the theories of the creation and the development of organic beings with remarkable skill and force.
Brief remark here. In Darwin's time, the theories of creation, what he's probably talking about is the biblical theory of creation, called creationism, which postulated that the species were made by God and placed on Earth all at the same time. Darwin is proposing that that's not what happened, that they evolved. Now, in Darwin's time, the creationist view was a tenable scientific theory. So Darwin had the burden not only of putting forth his own theory of evolution, but of competing with creationism. Today, of course, in science, creationism has largely been removed, or at least diminished in importance in biology. So that's a burden we don't have to face. Does anybody believe in creationism? Okay, I would say the following, or I will hypothesize that some of you do believe in creationism. Okay, that is, if you grew up wherever you grew up, if you happen to be in a Christian family and one that was devout, you might have learned about the creationist story of our coming here, right? The Genesis. And you probably went to school. I assume you all went to school, and you probably got educated in science and all this stuff. And there you acquired a contrary to view of where we all came from, and that's what we could call the scientific view. And that view does not lend credence to creationism. Okay. So I would suspect that many of us have two different ideas. Ones we picked up probably when we were younger, and ones we picked up subsequently. One that suggests that the biblical account is correct, and the other that suggests it's not correct, and that there's a scientific account, evolution, for example, that explains it better. Okay, so I believe lots of people have both these things. Our brains harbor contradictory beliefs all the time.
Author, 1855, has also treated psychology on the principle of the necessary acquirement of each mental power and capacity by gradation.
Okay, can you guys hear me now?
Yes.
Excellent. Okay. Yes, so I put a footnote on psychology there, or an endnote marking. What I'm about there is I have a lot to say. That is, the theory we're going to expose in this course has a lot of implications for psychology, but you won't find much on that in DTD, Darwin, Turing, Dawkins, or in this document. But I think it would be fun and good to discuss the implications of the theory that's being laid out here for psychology. Okay. Now back to the document.
Spencer's work on the evolution of societies received mixed reviews from other prominent sociologists. Spencer's concept of social evolution provides a useful framework for understanding the development of societies. Max Weber. Spencer's theory of evolution is a valuable contribution to sociology, even if it is not entirely accurate. Emil Durkheim. Spencer's theory of evolution is a reactionary ideology that justifies the status quo. Karl Marx.
So I put a footnote there just because I found Karl's comment kind of amusing. So Max Weber and Emil Durkheim are considering Spencer's theory as scholars. They're making a evaluation of its value to sociology. On the other hand, that's not what Karl's doing. So Karl Marx is more or less making an ad hominem attack of Spencer's work, right? He's saying it's a reactionary ideology that justifies the status quo. It's kind of funny that basically he seems to be calling Spencer a tyrant, right? Or some words to that effect. Anyway, not an important point, but I was amused. Okay, so let's continue.
Over a century has passed since Spencer and Darwin completed their work, and in that time much has been learned. Equipped with this new knowledge, we will pick up the work that Spencer began and explore a new theory of evolution in the social sciences. Evolution.
Okay. So, evolution. All right. So, evolution. Why is Darwin famous? Maybe some of my questions are so general you don't know where to start, but why is he famous? Anybody?
He disseminated a theory of evolution.
Yeah, except, as we'll soon see, Ryan Rubel, he wasn't the first to do that. Okay. Nor was he the first to observe evolution in nature. Okay, so let's continue.
Darwin was not the first to observe evolution in biology. In his works, Darwin cites thinkers going as far back as Aristotle, that in one way or another observed evolution and attempted to explain it. In On the Origin of Species, Darwin cites Lamarck, who in 1802, 7 years before Darwin's birth, had published Recherches sur l'organisation des corps vivants, wherein the theory of evolution now known as Lamarckism was put forth.
Okay, so even before Darwin was born, people, an example being Lamarck, were aware of evolution and trying to find a theory for it. Okay, so what do you guys know about Lamarckism? Anybody know anything?
So I think Lamarck, what he said is that changes that happen within the life of an organism are passed to the descendants, but that's largely not true, as I understand. So
Yeah,
like
so
a giraffe, because they try to reach for higher and higher branches, that will make their neck grow longer. That will pass to the descendants, but that's not really quite how evolution works.
right. So yeah, Jose Luis Ambite is correct. Okay, so Lamarck, just to reiterate what he said that, the giraffes keep stretching to get higher and higher on trees where they can find food, and that this made somehow their necks longer, and that lengthening was passed on to their offspring. Okay, so Lamarck was no joke. He was a very brilliant guy, and it just so happens that his theory didn't hold up. All right. So,
I want to
Unfortunately, Lamarck's theory.
sorry?
Sorry, I wanted to just add to what Luis said, that in one generation, it's hard to transmit the information, like whatever they learned in the next, but it's quite not correct, right? There's some evidence that the gametes are selected or passed on to the next based on the current pressures on the individual.
Yes. So, Sampad Mohanty. Yes, you're right. And I'm glad you mentioned it because I was remiss in not mentioning it. There is a topic in biology called epigenetics. And I think that's what you're talking about. And it does have some Lamarckian qualities.
Yeah.
Yeah. So,
Genetic markers to get passed to the offspring, and that's going to get acquired. But they're not permanent like in DNA. They can disappear after a few generations.
no, you guys are right. Okay. So I totally agree. However,
Unfortunately, Lamarck's theory was wrong in important respects and insufficient in others.
okay, so that's what happened. So,
What Darwin contributed was the first theory of how and why evolution occurred that could pass scientific scrutiny.
so that's what Darwin did. He came up with another theory of how and why evolution occurred that could pass scientific scrutiny. That is, was accepted by the scientific community and has not subsequently been dropped by the scientific community. Okay. So,
Beginning with Herbert Spencer and Auguste Comte, many social scientists have taken up the challenge of developing theories of social evolution. Among the early thinkers whose works touched on the topic were Lewis Henry Morgan, Edward Burnett Taylor, Talcott Parsons, and Immanuel Wallerstein.
are any of you social scientists? I think that there was a woman at the first lecture who was interested in media and maybe might consider herself a social scientist. Okay, I am not a social scientist, of course, and but I mention all of these people because they're famous social scientists, and this document is being written, for them as well as for you guys. All right.
In On the Origin of Species, Darwin had laid out the fundamentals of biological evolution, variation, the birth of individuals that were not identical to their parents, and natural selection, survival of the fittest.
Okay, so let's talk a little bit about Darwin. Hang on a second. So, how many of you have actually read, and by read I mean listen to, however you got it, The Origin of Species? Now, unfortunately, I've only read part of it. I'll finish it sometime. When I first started to read it long ago, I couldn't follow it because you had to know a lot about, raising cattle in England in the mid 1800s, or raising pigeons or stuff like that. And, I knew nothing about those things. But I've listened to it time and again now, and now it's making more and more sense to me. And one thing that I can say about Darwin is he's another guy that was no joke. That guy was smart. Um, yeah, just brilliant. And he argues his case for his theory of evolution brilliantly. Okay, so I'll mumble a little bit about what he did. He noticed that there was a variation, that individuals were typically not identical to their parent or parents. If you become a parent, you expect your child will not be identical to you nor to your mate. And they're variants. Okay. And then he noticed that there was such a thing that he labeled artificial selection. That if you were a farmer, and let's say you were trying to raise pigs, that you made more money if you raised bigger pigs. So you would start to, when I don't know whether you call them a litter of pigs was born, you might select those that came out big and keep them around to build the next generation. Whereas the ones that didn't come out as big, you might just send to market. Okay. And that it was quite clear that through this artificial selection, pigs on farms became bigger and bigger and bigger. They were selected not by nature, but by the farmer. And so he figured that variation existed all over nature, and that there might be an analog of artificial selection, which he labeled natural selection. And the argument was that nature itself was selecting those that were most fit for whatever nature's criteria were, not the farmers. And that's sort of basically some of his stuff. Okay, so at this point, we're going to turn to a review of biology. Many of you aren't biologists, so this review will bring you up to date on biology. And perhaps many of you have taken biology in school a long time ago and have a view of the cell as basically a vessel that contains a chemical soup where molecules run around at random and interact with one another. The first video we're going to see should disabuse you of that notion. The cell is far more complex, and rather than thinking of it as just a chemical reaction taking place in a closed vessel, it's better to think of it as a computational device carrying out an algorithm. So let's watch the first video now.
What I am going to show you are the astonishing molecular machines that create the living fabric of your body. Now, molecules are really, really tiny. And by tiny, I mean really. They're smaller than the wavelength of light, so we have no way to directly observe them. But through science, we do have a fairly good idea of what's going on down at the molecular scale. So what we can do is actually tell you about the molecules, but we don't really have a direct way of showing you the molecules. One way around this is to draw pictures. And this idea is actually nothing new. Scientists have always created pictures as part of their thinking and discovery process. They draw pictures of what they're observing with their eyes through technology like telescopes and microscopes, and also what they're thinking about in their minds. I picked two well-known examples because they're very well known for expressing science through art. And I start with Galileo, who used the world's first telescope to look at the moon. And he transformed our understanding of the moon. The perception of the 17th century was the moon was a perfect heavenly sphere. But what Galileo saw was a rocky, barren world, which he expressed through his watercolor painting. Another scientist with very big ideas, the superstar of biology, is Charles Darwin. And with this famous entry in his notebook, he begins in the top left-hand corner with, "I think," and then sketches out the first tree of life, which is his perception of how all the species, all living things on Earth are connected through evolutionary history, the origin of species through natural selection and divergence from an ancestral population. Even as a scientist, I used to go to lectures by molecular biologists and find them completely incomprehensible with all the fancy technical language and jargon that they would use in describing their work, until I encountered the artworks of David Goodsell, who was a molecular biologist at the Scripps Institute. And his pictures are all, everything's accurate, it's all to scale. And his work illuminated for me what the molecular world inside us is like. So this is a transection through blood. In the top left-hand corner, you've got this yellow-green area. The yellow-green area is the fluids of blood, which is mostly water, but it's also antibodies, sugars, hormones, that kind of thing. And the red region is a slice into a red blood cell, and those red molecules are hemoglobin. They are actually red. That's what gives blood its color. And hemoglobin acts as a molecular sponge to soak up the oxygen in your lungs and then carry it to other parts of the body. I was very much inspired by this image many years ago, and I wondered whether we could use computer graphics to represent the molecular world. What would it look like? And that's how I really began. So let's begin. This is DNA in its classic double helix form, and it's from X-ray crystallography, so it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring the right way inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you. This is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding soup of molecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of its field of view is a single strand of DNA. This huge package of DNA is called a chromosome, and we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science, at the frontier of human understanding, is mind-blowing. Discovering this stuff is certainly a pleasurable incentive to work in science. But most medical researchers, this is just discovering this stuff is simply steps along the path to the big goals, which are to eradicate disease, to eliminate the suffering and the misery that disease causes, and to lift people out of poverty. Thank you.
That was an amazing video. And it illustrates what an incredible extravaganza it is of choreography to get the cell to divide. A remark about this. Consider like a Rolex watch. I mean, inside its case, there are about 200 parts. And it is a mechanical device carrying out an algorithm. It'll show you the time, it'll show you the date, all those things. The smallest of its pieces is about 20 millimeters. Yes, I think that's right. And that is about 1,000,000 times bigger than the pieces you just saw in that video, like those little robot-y molecules walking down strands. Um, Not only are the pieces smaller, but the watch has like 200 or so pieces, and they are intricately put together and work in just the right way with one another to make this watch carry out its algorithm. But in the case of cell division, there are millions of these little pieces, perhaps even billions of these little pieces that are doing this incredible thing. That is much, much finer, much more intricate, much more complex than a Rolex watch. So nature is amazing. And it should make it clear that what's happening inside the cell, while it can be thought of as just chemical reactions, and if we step back to a more reductionist view, it can be thought of as just the activity of physics. But when you see that video, you realize that what it is an extraordinary computer that is executing some algorithm. And that's the take-home message of that video, for our course at least. All right, let's turn to the second video, which addresses something called the central dogma of molecular biology.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
The last video resulted in a discussion of the nature of molecular computation, DNA computing, and some topics in theoretical computer science, including Turing's definition of a universal computer. If these topics are not of interest to you, then feel free to just skip this part of the video. Now, I assume you're all familiar with the fact that A's only want to bind with T's and C's only want to bind with G's. Yes? Seems pretty basic. Okay. Now, by the way, just as an aside, if any of you are familiar with my work in DNA computing, I had reason to believe to start reading a book on molecular biology about 30 years ago. And when I read about that polymerase that runs down the DNA and makes this new strand of RNA with the same information as was on the DNA, I remember it's one of two times, the other time involved this course, when I suddenly sat up and I said, oh. And what I sat up and said, oh, about was, how many of you know what a Turing machine is? Have you encountered Turing machines? Yes. I mean, if you're a theoretical computer scientist, you should have. All right. That polymerase running down information and writing a new chunk of information was like a Turing machine running down a tape of input and producing a tape of output. And I said, when I saw that, I said, oh, these things in biology, these molecules can compute. And that's what led to that particular path of DNA computing. All right. So let's see. If you watch that video, I'm sorry. Who spoke?
Oh, Nathan or Hockey.
Oh, Nathan. Hi.
Hi. I
Yes,
was
did
just
you want to say
wondering
something?
for the analogy of the Turing machine versus RNA reading, how are there different states in RNA, or is it just each state's a different amino acid?
Ah, what a great question.
Do they have enzymes?
Yeah. Huh?
Do they have enzymes that kind of turn off and on certain things for RNA? So it's kind of like
Yeah, they do. So how is it like a Turing machine? And does it store state? Well, I'm talking about the enzyme. Enzyme's a word for protein that does something, catalyzes some reaction. And the enzyme of interest is a polymerase. Okay. In this case, it was what's called an RNA polymerase, but there are others. Okay. And this molecule runs down it jumps on to the DNA and it starts to walk down the sequence of A, T, C's, and G's. And while it does that, it's reaching out into the surrounding solution and grabbing little RNA molecules that it's putting in a system. So what it does is when that polymerase is reading a particular base, it changes conformation, as it's called. It changes its shape a little bit so that now it will only accept one of the RNA bases that matches it. And it'll add that to the strand. Now it'll move down another step on the DNA and see what it's reading there. and it'll change its shape a little differently than it did before if what it's reading now is not the same DNA base. And so that it will only accept the right RNA molecule to add to its chain. So yes, the polymerase does have state. Good question, Nathan.
Thank you. That's an interesting explanation. Thank you.
Yeah. Okay, it does have state. And now it is
Sorry,
also the
I kind of work something a little bit on genetics. I'm not sure if I understood the answer, the question. But you're talking about translation from DNA to messenger RNA, or translation from messenger RNA to protein in the ribosome.
Oh,
The question.
okay.
I
By the
mean,
way, can you raise your hand so I know who's talking? I'm looking at all of these.
so-
You know, use the thing to rate.
yeah,
Jose,
yeah.
Luis.
Again, sorry.
and-
Like I work on
No.
genetics. I work on genetics and on neuroscience and on a bunch of things. But I mean, if the question was if these things have a state, I don't think they have a state. They just kind of they run over the sequence and they translate. No, if it goes from either DNA to messenger RNA, and that's like a one-to-one base translation, and then the messenger RNA gets out of the nucleus and goes to the ribosome, and there for every 3 nucleotides, then you get an amino acid attached. But it's kind of reading a tape. I don't think there's any type of a state. It's just translating or transcribing what it's reading. I mean, what's the question? I may have misunderstood the question.
Okay, so let me try to clarify what I think is going on. Okay, there are these molecules called polymerases. All right, and this is for translating the information in a DNA strand into an RNA strand. Okay, the same information, just in a different molecule. Yes. And the question that I took Nathan to be asking is, well, if I saw this as an act of that looked like Turing machines to me, what I saw was that there was this finite state machine, this polymerase, and that it was reading a tape that had a bunch of A, T, C's, and G's on it. And it was writing a new tape, which had a bunch of, A, T, C's, and
U's.
Is it U's? Yeah.
Brazil.
Yeah. And so it was reading a tape and writing a tape. And I claimed that reminded me of Turing machines. And Nathan asked, well, if it's really a Turing machine, then that enzyme is the finite control. Does it really store state? And I claim the answer is yes, because if it's reading an A, it will undergo a conformational change as a molecule, in which case, in the surrounding solution, only an A, well, it might be a T, I don't know, only the right RNA base will now stick to it and get added to the chain that is being grown. And now when it moves one step to the right, it changes state depending on what the next nucleotide in the DNA is. And now we'll only accept the corresponding RNA molecule. So it stores state in its conformation at any particular moment. It has sort of one of 4 states, and it switches to them depending on the input. All right, that's number one. That's my interpretation. Now, Jose Luis, you are also talking about the ribosome. Now, the ribosome is a real big complex that involves RNA molecules and lots of enzymes and lots of proteins, and becomes this package. and very complex. But it too is a computer. In this case, it's reading the information on an RNA strand and writing information onto an output strand that is amino acids that are going to form the growing protein. Now, does it have state? It definitely does, because when it reads a, what do you call a 3 in a row on an RNA? It's a codon. When it reads a codon, it switches states and so that it will only accept the right protein. Now, really, I guess we could argue about whether it's the transfer RNA that's part of the ribosome or not. I don't know. Anyway, yes, these molecules do store state. It's a little more complicated for me to describe it because I'm a little unsure in the case of the ribosome. But I would say in general, yes, they store state. And okay, so that's what I see, Jose Luis. What is your take on that?
No, I mean, I think we're splitting hairs. I mean, to me, they're very simple machines. I mean, they're not simple, they're very complicated molecular machines. But nonetheless, the function they're doing is something quite simple. They don't have like long-term memory in a sense. They just kind of react to, okay, I see an A in DNA, I'm going to generate an A in RNA. And I see,
yes,
I always forget, a G, I'm going to generate a G. So it's kind of a simple thing. And in the ribosome, it's a bit more complicated, but you have 3 letters. And there's a code, there's 64 combinations for 3 letters, and there are only 20 amino acids. So there's like a table that tells you from which 3 letters, which amino acid you get. But again, it's not remembering a code on, I don't know, 20 steps before. It's just doing it completely one after the other. So that's why I said
totally agree,
state, but not a long-term state. I think it's only just translation.
Oh, I'm sorry. Okay, I
I
totally
don't want to
agree
take more
with you.
time. Maybe we're splitting hairs here.
No, I want to take more time because maybe we could clear this up by asking, well, if these things really compute, as I'm claiming that, even the simple things we've looked at are little acts of finite state machines on tapes, then how many of you are familiar with the notion of a universal computer? Okay, Isabel, Lee is. Do you guys know what that is?
Is that just something
Does
that could
everybody know?
simulate a Turing machine?
Okay.
Or
who's talking?
oh,
Sorry.
Nathan Derhockey. Sorry, I could raise my hand. Was that the thing that could simulate a Turing machine? A universal computer? Or is that something else?
Okay. A brief detour into theoretical computer science. Is an abacus a computer? There were all sorts of machines, an adding machine, a computer. Okay. And you could say, sure, it computes something. But what Alan Turing, yes, did in 1936, I think it was he cleared up what a computer was going to be and what it wasn't going to be. All right. Now, if you take a computer today, you load it with a program and you load it with an input and it does some calculation, right? Now, what kind of things can be computed? Is there any notion of when you write a program, what it computes? Well, it computes something. Depending on the inputs you give it, it'll give you outputs. In the eyes of a mathematician, it's a black box. that receives arguments and gives values.
And
right.
Turing machines are based on the assumption that these things that it will be computing is enumerable, correct?
Are what?
They're enumerable?
Yes, right. You're certainly saying that something that I think is the right thing. Now, what Turing said was the collection of all the things you can compute with all the programs that you can write is actually a set which I'll give a name to, and the name he gave, I forget what he gave, but it later became recursive functions or whatever. Okay, he said it's a set and we can make sense of it. And the beautiful thing about the Turing machine is if I gave you one Turing machine and enough tape, then you could program it with different programs that are Turing machine programs. And every function that is computable by any kind of computational device, there'll be a program that computes exactly that same function on a Turing machine. And he said that makes Turing machines special because they can compute informally. They can compute everything that can be computed. That made them universal machines, he called it.
And Professor, correct me if I'm wrong too. Even though they can compute all of that, his paper came to the conclusion that there's a finite amount of things that it's able to compute, correct?
No, there's a infinite amount, and for what it's worth, if you don't know, it's one of the great results in mathematics, my opinion, that there are different sizes of infinity. Okay, we won't get into it now, but what Turing showed is this was what's called a countably infinite set. It's infinite, the number of functions that can be computed on it. And given a Turing machine, it's not only infinite, but his claim was that you could never build any kind of computational device in our universe that could compute a function different from this set than the things in these in Turing's set. Okay. Abaci can be used to compute a lot of stuff, but they're not universal. You can't, for example, make them tell you whether a number is prime or not. Okay. Okay, so there's this notion of universal computer. So now the question that I think Jose Luis was getting to, we can ask this question to perhaps answer that. And this question is, are the molecules in the cell, can I use them as a universal computer? That is, if you give me all the molecules that we find in the cell, and I can use them any way I want, that is, pour them in a little test tube, as many of each as I want, then can I in fact compute everything that could be computed on a Turing machine or on your iPhone or on your desktop? Is it universal? Is that a universal collection of primitive operations, one could say? And anybody know the answer?
I would think no, because one of the assumptions of the Turing machine is that it can change its next action based on the things that it's read, which I think the polymerase cannot.
I'm not limiting myself just to the polymerase. I mean, you know, I might use a ribosome here, and I might use DNA.
Okay, I don't have much bio background, so the distinction doesn't really mean much to me.
Okay, that is, there's a treasure trove of molecules that have evolved over time, and they do interesting things like the ribosome. So can I use those molecules to compute anything I want to compute? And I know that's vague, and it's a lot to take in, especially if you don't have all the background that this refers to. But the answer is yes, universal. Okay. So that's the sort of an answer to your question, Jose Luis, right? Those tools, those little computers, we just looked at one of them, polymerase. Yes, you can use those to build a universal computer. Okay.
Sorry, I
And
had a
that
quick
computer,
question.
yes.
Are we distinguishing, so like, are we saying like the genetic, like the components that are like currently within our body right now, like I think we're talking about the ribosome and the polymerase, those specifically can be used to implement the, I know we're calling it just a general Turing machine. I assume it'd be referring to like a universal Turing machine, right?
Yes. Here's what I'm saying. That let's say you want to compute tell whether a number's prime or not.
Mhm.
And you want a function. I mean, you want to build an algorithm that I can put in any number I want, and it'll tell me whether it's a prime or not.
Yeah.
Okay. Then I can, if you want to do that, then you tell me your number that you want to know is prime or not. And I can make a mixture in a little test tube.
Mhm.
And if your number was prime, and I'm going to encode your number in a strand of DNA and drop that in the test tube. If your number's prime, then guess what? That tube will turn red. And if it's not
Mhm.
prime, it'll turn blue. That is, I know how to make that mix of chemicals that'll have that behavior. Okay, so this is a whole area and, kind of very interesting.
Can I ask a question on this?
Yeah.
And I guess I have my limits on theoretical computer science and Turing machines.
Yes.
But
Yeah.
so I thought that, well, I think the original Turing machine, I think you needed an infinite tape, but I may be wrong. Maybe there's something with finite tapes. But I think there are other things that are equivalent to Turing machines that, like production systems or rule-based systems that can evaluate any. Because I do buy. I mean, I'm trying to push. I know that you already did a lot of algorithms on DNA, so I know your previous work, but it doesn't mean that a biological system is Turing complete in the sense that you can do any function. Because I mean, maybe just for people that don't know too much biology. So in the DNA basically there's something that triggers the polymerase to attach to some part of the DNA and then transcribes like a gene. That gene kind of comes out of the nucleus and then it's transcribed to a protein. That protein will cascade and interact with other proteins, some of which may come back to the DNA and trigger another transcription. So I can believe intuitively that that mechanism can produce all kind of proteins. But there's a finite kind of, well, there's a finite space type of thing. There's only so much DNA that you can do. What I see as maybe infinite and be able to simulate any computation is this kind of recursive process that you can generate a number of proteins that do some things and then go back to the nucleus and or to the tape and generate other proteins and that can go. But I still not sure if that's truly Turing complete. But you probably have thought more about that and understand this better than I do. And it's a question.
Yeah. So what if I limit the amount of memory that you can have in your computer, RAM and on your drives, but I limit it. You only get to use what's there. Then how many functions do you think you can compute? Not much. Okay. In order for us to think that you have a universal computer, we also have to think that you have sort of unlimited memory. That is, if you run out of memory, you can put more in. And that's true of DNA and all other computational devices. If you really put them in a box, and you don't let any more memory in, then you can't, for example, tell whether numbers are prime or not. So anyway, these are great questions, but I think we should pass on them right now and leave it where it is. And, you know, we got to go on, but they're great questions. They're very deep questions, and they can be answered.